Hello everyone and welcome to this new Gen webinar sponsored by Horizon Discovery entitled Cell Lines for Optimizing Biotherapeutic Production Workflows. I'll be your host today. I'm Jeff Galiskis, Technical Editor for Gen, which is going into its 40th year now, covering the discovery and innovations within the life sciences industry. Gen has been around to see some of the earliest uses of animal cells for the production of therapeutic proteins, which has been an integral part of the biopharmaceutical industry from the beginning, with CHO cells being one of the cornerstones for their success. Comparative to other mammalian cells, CHO cells have an amazing adaptability to all types of cell cultures, like bioreactors for instance, which makes them an ideal choice to produce one of the most successful protein therapeutic classes, monoclonal antibodies. In recent years, cell line engineering efforts have generated improved host cell lines for therapeutic production that maximize protein expression and increase their robustness in bioproduction. A prime example of this is Horizon Discovery's CHO-Source GS knockout cell line, which genetically disrupts the glutamate synthase gene, facilitating the selection of high-expression clones with optimal performance during cell line development. Today, we're going to see how this cell line is being used by Elanco Animal Health to build a biotherapeutics research program. Now, it's my pleasure to introduce our presenters for today's webinar, who are going to tell us more about this unique cell line. Dr. Mario Pereira is a field application scientist in bioproduction for Horizon Discovery, and he'll begin the presentation today by briefly telling us some more specifics about the CHO-Source GS expression platform and how Horizon Discovery can assist your biotherapeutic production efforts. This will segue nicely into our main presentation from Dr. Alan Spear, who is a principal research scientist for discovery research at Elanco Animal Health. Alan will tell us his experiences with the CHO-Source cell line, focusing on the processes for evaluating growth conditions, transfection methods, clonal cell line development, and productivity assessments. But before we begin, I want to remind the audience to submit questions for our Q&A session that is going to occur right after Alan's talk. Don't be shy, send in your questions at any time for Alan or Mario. All you need to do is click the Ask a Question panel on the right-hand side of your screen, type in your question, and hit Submit. We'll try to get to as many of your questions as we can. Okay, with that said, Mario, I'm going to turn the presentation over to you.
I would like to start to thank you all for attending this webinar today, and also to thank Alan for accepting being our first guest speaker. My name is Mario Pereira, and I'm a field application scientist here at Horizon, and my main role is to support our CHO-Source customers during the implementation of this platform, as well as after this platform is up and running at their sites. I don't want to spend too much time today before Alan's presentation, and for that reason, I only have 4 short slides to quickly introduce Horizon to those customers that are not very familiar with the company, as well as to introduce CHO-Source platform and our gene editing capabilities that are focused on the bioprocessing market. I hope at the end of today, you'll be able to see that CHO-Source is a very well-established platform, is suitable for manufacture of different types of biologics, and with Alan's presentation, you will be able to see that overall performance of this platform in customer hands. Horizon is a products and service biotech company that supports science and scientists during their drug discovery process, all the way from research to therapy. We do this based on our 2 technology foundations that are gene modulation, with, for example, RNA interference technology, and gene editing, with all our CRISPR-based products and services. Based on these 2 technology pillars, we are divided in different units to support our customers with research reagents, diagnostic reagents, screening and product services, and bioproduction products and services, where we have our CHO-Source platform that we are introducing here today, but also CHO cell gene editing and CRISPR screening services. Our CHO-Source platform foundation is in our HD BioP3 cell line. This is a well-established CHO-K1 based cell line in which glutamine synthase was knocked out using our proprietary AAV technology. We also sublicense a non-transposase dual promoter expression vector from Atom for those customers that do not have their own vector of choice. This vector is an hybrid dual promoter vector that, depending on the cloning strategy used, it allows both cloning of dual chain antibodies as well as single chain molecules. On top of that, we also have a comprehensive set of optimized protocols for you to work with this cell line. These include all the instructions needed on the generation of the final protein expression line, from transfection, selection, single cell cloning development, as well as clone characterization. Finally, we also have a full cell line history package that contains all the traceability documentation regarding the development and manufacturing of HD BioP3 cell line that will support you on your regulatory submissions. ChoSource was introduced in the market in 2016, and since then we have licensed this platform to more than 80 customers. Some of our customers, like Elanco, are involved in animal health sector, but the majority of them are human health biotech, biopharma, and CMOs. Until date, we have 7 disclosed INDs, 6 in US and 1 in China, and these are on different type of molecules, from the typical monoclonal antibodies as well as to the less common bispecific and Fc fusions, and also focus on different disease areas, from the infection disease, oncology, as well as autoimmune disease. Finally, I just want to give you a brief introduction to our gene editing capabilities. For the last few years, we have developed a set of gene editing tools and protocols optimized for CHO cell engineering. Due to the IP restrictions around CRISPR, we typically use CRISPR for R&D projects, like proof of concept studies, and then we use recombinant adenovirus technologies to develop our gene editing cell lines to be used in a GMP context. With both technologies, we can generate a variety of genomic modifications, such as knockouts, knock-ins, point mutations, deletions, landing pads, and others. As part of our set of tools, we also developed an all-genome CRISPR library optimized for CHO cells. This allows us to offer our CHO CRISPR screening services, where, in an agnostic manner, we can work with you to identify which genes are potentially involved in bottlenecks that, for example, affect your expression and product qualities on your current biologic molecules. To end, I want to thank you all again for attending this webinar today, and to let you know that if you have any questions about bioproduction products and services here at Horizon, please contact us on bioproduction@horizondiscovery.com. Finally, on behalf of Horizon, I want to thank Alan and Elanco once again for their time and customer testimony on their experience with our ChoSource platform.
Thanks, Mario. Great start. Real quickly, before Alan begins, I just want to remind the audience once again to stick around for the Q&A session and send in your questions for Mario or Alan at any time. All you need to do, again, is click the Ask a Question panel on the right-hand side of your screen, type in your question, and hit Submit. Okay, let's get back to the presentations. Alan, the Gen audience is all yours.
Well, I'd like to start off by thanking Mario and really the whole team at Horizon Discovery for providing me an opportunity today to speak to you all. My name is Alan Spear. I am with the Discovery Research Group at Elanco Animal Health. I'd like to talk to you about our experience using the Horizon Discovery ChoSource cell line as we built out our capabilities in animal health biopharma. So I will start with an introduction to Elanco Animal Health, sort of who we are and what we do, and then give a very brief overview of animal health biopharma for those of you not in animal health, and then get into sort of how we got started with the HD BioP3 cell line, the ChoSource cell line, sort of standard culture and media, move into our experience in testing and optimizing our transfection, selection, and integration processes, and then follow that on with our platforms for single cell cloning, sort of our CLD workflow, and then what we've done so far in terms of titer testing and scale up of therapeutic expressing clones from the ChoSource platform. So Elanco Animal Health is really a purpose-driven company. Our core value of food and companionship enriching life is really embodied in the statement, making their lives better makes life better. And I know that over the past year, as many of us have become more and more disconnected from each other, those of us who were already pet owners and many people who became pet owners have truly understood the power in that statement and truly understood the value of that human-animal bond. So many of you may not be familiar with the name Elanco. We were actually founded in 1954 as a division within Eli Lilly and Company, which is actually where the name comes from. The Eli Lilly and Company, all condensed to Elanco. We are headquartered in Greenfield, Indiana, just outside of Indianapolis. We have about 10,000 employees globally, 20 manufacturing sites, and our combined pet health and farm animal revenue in 2019 of $4.6 billion. We represent about 200 different brands. We're selling products in 90 countries, and we have grown significantly over the last 10 years, both organically as well as by acquisitions of multiple other smaller and larger animal health companies. In sort of a timeline form, you can see like I said, we were founded in 1954, and so we actually consider ourselves a 66-year-old startup company. While we continue to grow and change and shift our portfolio as a part of Eli Lilly and Company, including acquisition of a vaccine portfolio and Novartis Animal Health in 2000s into 2014. We actually became a publicly traded company almost exactly 2 years ago, and fully separated from our parent company, Lilly, in 2019. And very recently, our largest acquisition and actually the largest acquisition in the animal health space, Elanco acquired Bayer Animal Health, only finalizing that acquisition 2 months ago. And at the time, Elanco was the fourth largest animal health company globally, and Bayer Animal Health was the fifth. So we are all very excited about the added possibilities, the added scope, the added potential of this combined company going forward. So as I said, I want to give you all a brief introduction to animal health biologics. In no way am I qualified to speak for all of animal health, but given what I believe to be the primarily human pharma background of most of the attendees, I wanted to at least give you some points of reference. So really, sort of before about the year 2000, a biologic in animal health largely meant a vaccine. And even today, 20 years later, there's only one canine monoclonal antibody, or really only one animal veterinary monoclonal antibody on the market, and that is a canine monoclonal which targets IL-31 to block itch signaling in atopic dermatitis. And actually, it's not only on pace to become an animal health blockbuster. It truly is an animal health blockbuster, having done over $100 million in revenue in 2018 and I believe in 2019. So the blockbuster status in animal health is about an order of magnitude lower than human pharma blockbusters. So it's really opened up the potential for biologics, for true biologic therapies in animal health. And to that end, we anticipate multiple market entries from multiple animal health companies, primarily in the therapeutic areas of dermatitis, pain, and oncology. Interestingly, in the U.S., we have a split regulatory path. So There's the USDA, the U.S. Department of Agriculture, Center for Veterinary Biologics, and then the Food and Drug Administration, the FDA, Center for Veterinary Medicine. Now, these two agencies do have a memorandum of understanding. However, the pathway, the regulatory path for biologics is actually contingent on the mechanism of action. So I won't go into all the details, but it does present a rather unique challenge to those of us who may be involved with some of the sort of documentation and regulatory requirements, having to go through one or eventually really both of these pathways. Outside of the U.S., the regulatory paths are more straightforward, sort of by region or by country. Really, as I mentioned before, the only current monoclonal antibody therapy is in dogs, and the current focus is largely on companion animals, so pets, and that's primarily due to cost of goods. So the increased cost of manufacturing a biologic drug really cannot at this time be borne out by the farm animal space, by the margins that are sort of permissible in that space. Now, I'm optimistic that developments will be made on multiple levels that will change that dynamic, and I'm very excited to see the direction that we might be able to go outside of dogs and cats. But as of right now, and I would anticipate in the near term, that will be the focus of sort of animal health monoclonal antibodies specifically. So really kind of from here on, I'm going to be sort of walking through, I guess, our process to build out ultimately from scratch a mammalian cell bioprocess, sort of biotherapeutic research workflow, and one of the foundational decisions that we made was to license the CHO source, the HD Bio P3 GS knockout cell line from Horizon. So upon starting our evaluation of the cell line, we first just asked, can we get these to grow? And we initiated our work with the CD4 media, and that actually continues to be sort of our standard media for culture applications. We also utilize the Efficient Feed C plus AGT supplement for fed batch runs. We are currently performing an internal effort to develop a custom media package with an eye on cost of goods. Our initial focus is on developing a production media. However, we have a sort of a long-term aspirational goal to have a single all-purpose media and feed package that will support everything from sort of early research, transfection, CLD, all the way through early process development and into production. We are not there yet, but we believe we will get there. Based on some growth curve experiments that we did early on, we've sort of established our standard maintenance passage at seeding at 0.2 to 0.3 million cells per mil on a sort of standard humidified CO2 incubator, 125 RPM, 25 millimeter throw. Under these conditions, we generally have seen the cells start to top out of exponential phase around 7 to 8 million cells per mil, and so we do not like to exceed 4 to 5 million cells per mil in routine passaging. We have found that keeping cells within this range, as I'm sure many of you have already experienced, really keeps them growing steadily, optimal conditions for transfection, and we've seen within this range, doubling times of 18 to 20 hours. So the cells are very robust. They grow very well under these conditions. And then just as really just as an aside, we've utilized both the Nova Bioprofile Flex Analyzer as well as the Beckman Coulter ViCell for automated cell culture monitoring, cell counts, viability measurements, and the cells perform relatively well on both of those instruments. So we're able to use either one for different purposes during our routine culture or during fed batch runs. So moving into our transfection selection and integration workflows, we have tested a variety of transfection reagents and systems, including but not limited to the Neon system from Thermo Fisher, as well as the Nucleoffector from Lonza, and then multiple lipid-based transfection reagents, including the Freestyle Max from Thermo and the Transit Pro from Mirus. We have generally found the most success with the Freestyle Max and then the Neon reagents. However, the variability that we see in transfection efficiencies and post-transfection viability is also significantly impacted by our DNA vector size, the concentrations that we've used, the exact cell health and densities that we've used. And so really sort of our system has been optimized again for our vectors in our system, the DNA concentrations that we've optimized for our particular plasmids. All found that primarily to be optimal in the Neon transfection system. That does tend to give us the highest efficiency and highest post-transfection viability. What I will say is we are still currently building up our data sets in terms of not just sort of the immediate transfection efficiency and viability, but longer term, going through multiple pools in parallel, taking things through our CLD workflow, looking at our final titers and distribution of titers. And at this point, I don't know if I'd be willing to say that there is a very strong correlation between our initial transfection efficiency and viability, and then what our eventual titers are. As I said, our data set is not hugely robust right now, but I have been a little surprised that the correlation has not been nearly as tight as I would have originally thought. So going further into our selection processes, you may be thinking, well, the show source cell line is a GS knockout cell line, so it should be fairly straightforward. And that is true. We have successfully used multiple different selection methods. While the GS MSX selection is our primary method, and we routinely are getting pools recovering in 14 to 28 days, we have also looked at other antibiotic selections like puromycin or hygromycin B, G418. All of those still work in this system. You do have to add glutamine to your media, but the puromycin selection actually does work significantly faster, and in many cases we can turn around pools in a week and a half, no more than 2 weeks. And that's turned out to be, quite a good platform for us for making reagents, tool compounds, and things like that. When we are just looking for something quick to generate some material and move on. We really haven't used other selection methods very frequently. We simply tested them out to establish whether or not they were feasible. They sort of represent an intermediate timetable in between the GS selection and puromycin selection, and so unless we would move to a multiple selection marker strategy, they are really not used much in our shop. We do use transposase-based integration for stable expression. And we currently have licensed both the Demetra piggyback transposase and Adams Leapin transposases. our experience with the piggyback system really extends back to our time as a division within Eli Lilly, and so we have the most experience with that platform. We continued using that platform when we separated and licensed the ChoSource cell line. However, we have established the functionality of the leap-in transposase system, and furthermore, the team over at Atom has a great cell line development group that utilizes the leap-in system alongside of the HD BioP3 ChoSource cell line, and has established a very efficient workflow for generating high titer cell lines. So it's clear that system works quite well in this cell line, right alongside the piggyback system. We routinely obtain stable pools expressing 1 to 3 grams per liter in this system. The primary variability here has been either by type of molecule, so antibodies versus Fc fusions versus sort of globular or soluble proteins, and then by selection method, either GS versus Puro, and then the stringency of that method. We have seen on average higher titers with GS selection than we do with antibiotic selection, Puro selection in particular. So we have played around with stringency in terms of MSX concentration, Puro concentration. You can force the average, the titer averages up a bit in the pools by increasing stringency, but there is a point where you can just push the cells too far, and you're not getting much for your return. The recoveries take much longer, and we haven't at this point seen that much of a benefit by ratcheting up the stringency of selection, past what I would call sort of typical ranges. And really at this stage in terms of stable pools, we really haven't seen that added value to pushing the timeline out to try to gain a few extra milligrams per liter in productivity at the pool stage. So getting into our workflows for single cell cloning, we have successfully used limiting dilution and FACS sort of as a single cell printer with the Forticho media. We have tended to use clarified, filtered 10% conditioned media as a part of this to encourage cell growth. And we have established that the speed of recovery of grow out and the hit rate has improved through the use of other supplements, and those include the stem cell clonocell CHO ACF and the Sal Scientific Instagrow. So we do know that those supplements are they both work well with the ChoSource cell line. And we've tested those in both plate-based methods, the limiting dilution and FACS sorting. However, our primary workflow for single cell cloning for CLD is actually the Beacon platform. So for those of you who are not familiar with the platform, this is a microfluidics platform that utilizes something called optoelectric manipulation. This utilizes sort of what I believe is a very elegant combination of light and electronics to almost use an optical cage to handpick individual CHO cells and put them in their own little pens. And this clonal selection allows us to clonally isolate, grow out, and perform productivity assessments on up to 6,000 cells, clonal cell lines, essentially in 5 days. This is a picture of the Beacon instrument itself. It's about the size of a large vending machine. The chips pictured here on the left are the 4 OptoSelect chips that get loaded into the instrument as pictured here on the right. This is a positive pressure, almost bisafety cabinet-like environment. Each chip has its own fluidics. There are two 96-well plate incubators inside this system, as well as media and liquid reagent bays. And so this system really is an all-in-one, self-contained culture system that allows us to visually isolate clonal cell lines and provides photo documentation of clonality, as well as an inline productivity assessment. And so to give you an example, I'll run through some data from a sort of a proof of principle run that we did with the CHO-source cells on the Beacon platform as we began our process of optimizing our Beacon workflow. In this pilot run, we used a single chip. So each one of those 4 chips that I just showed you has a total of 1,758 nanoliter level pens. I think each pen is about 1.7 nanoliters. We used the commercial Forticho media, as I've mentioned, and we used a cell pool producing a canine monoclonal antibody. The cells penned well, so we got 1,510 single cells out of the possible 1,758 pens in the chip. That's actually a very good efficiency. You will get a certain amount of doubles from cells that are stuck together, cells that the instrument can't optically distinguish initially as doubles. And then you will have a certain number of cells that do get missed or empty pens, which actually are used as assay controls later on. The on-chip growth in this particular run was adequate. We had about 33% of the cells that grew out to greater than 5 cells at day 4. However, this particular run, we did not utilize any conditioned media. We did not utilize a load reagent that also helps in growth, and we did not perform any sort of voltage optimization. So there is a low voltage that's applied to the pens to enable us to manipulate the cells, and there is some optimization involved in getting that voltage right. So we have since this initial pilot run, we have improved our on-chip growth, and in most cases we're at greater than 70% of cells growing, 70% of singles growing out by day 4. This just gives you sort of a graphical representation of individual pens within the chip. So this is a randomized sort of cell count. Each one of these dots represents a pen. Cell count is on the y-axis here. So you see we have, by and large, 5 to up to 50 or so cells per pen on day 4. We grow those out again on day 5. You see that we're now, a couple of cells approaching 100 or slightly over 100 cells per pen. And you can not only see that they're clearly growing, but you can also see that the growth is generally not necessarily even. You have a distribution of growth rates in terms of doubling times that we've seen from 16 hours all the way up to 40 plus hours. And these are all measurements that you can make on this system, because the system will count the number of cells in every single pen almost as often as you'd like it to, and you can generate that data and calculate growth rates as well as productivity measurements for each clonal cell line in each pen. When we go to assess productivity of each of these cell lines, we perform something called a spotlight assay. This is a fluorescence-based assay that is based basically off of the difference of diffusion of a fluorescently labeled detection reagent and the decrease in diffusion of that reagent once it's bound to a monoclonal antibody being secreted by the cell line. this assay, when you subject this to sort of a false color here, the principle of the assay is essentially the lower the fluorescence intensity indicated by the darker blue are low or non-producing cell lines, or empty pens, or pens of cells that didn't grow. So in this particular case, you saw that we had quite a few empty pens or pens that didn't grow out. The brighter green are high-producing cell lines, and in this case, the fluorescence intensity from each of these pens is quantitated. We can isolate each of our top 12 clones. And indeed, when we take a look at the fluorescence scores from empty pens, we can see this distribution down here and compare that to the distribution of fluorescence scores from pens that contained greater than 6 cells. You can clearly see a shift upwards, increasing fluorescence scores, and then indeed we see our top 12 clones as essentially outliers of this distribution, which is really exactly what we're looking for when we're looking for high-producing clones. At this point, once we've identified these top high-producing clones, we can utilize the same optoelectric positioning technology that was used to grab onto a single cell and bring it into the pen to also take a cluster of cells out of the pen now that they've grown out and export that to a 96-well plate for expansion. So in summary, we were able to demonstrate a OEP, optoelectric positioning, single load. I know this is kind of hard to see here, but this is the channel of the chip. This is the load of cells into the chip. And then down here are single CHO cells that were individually boxed from the channel and brought in, placed into the middle of those pens. We've demonstrated that you can culture them on chip. This is an image from day 4 of that grow out, and you can see there are differences not only in growth rate, but in morphology of the individual cells, how they cluster together. It's actually very amazing to think that what appears as a fairly heterogeneous or fairly homogeneous mixture in a pool, when you look at them on the beacon, just how heterogeneous that mixture really is in terms of growth rates and morphologies and productivity. We've shown that we can perform a spotlight antibody productivity assay on the cells and then successfully export the cells on day 5. And what you see here is essentially we've lifted with this same sort of optoelectric system, lifted cells out of the bottom of the well, gotten a packet of cells up into the channel, and then this can be flushed out directly into an individual well of a 96 well plate. Moving into our titer testing and scale up. So what do we do after we get things off of the beacon, and how do we assess the productivity? We have performed side-by-side productivity or titer testing in both 125 ml shake flasks as well as 24 deep well V-bottom plates using nominal volumes of 25 mils and 3 mils respectively. We've performed actually a full 14-day fed batch in these. We wanted to get as close of an estimation to a more production-like system. And we saw comparable productivity between the deep well plates and the shake flasks, the 125 ml shake flasks. I'd say by and large, most of the productivity measurements were within 10%, but across the board, I would say plus or minus 20%, and for the most part, rank order also stayed the same. As for our actual scale up, so one of the wonderful things that we can do with this whole system, as I've described it, is generate high-producing pools and high-producing cell lines fairly early in the research path. And in animal health, we can go into our target species very early in research. So we are routinely doing PK studies, immunogenicity studies, and even some efficacy studies very early in research in our target species. And to support the material needs for these studies, we're able to utilize these high-producing pools and high-producing clones in 1.6 liter Thompson optimum growth flasks, which give us sort of a high volume given for a given footprint. In this case, we're seeding about 900 mils in one flask at 0.3 million cells per mil. We perform a temperature shift to 32 degrees on day 5 and are continuing to use the efficient feed C plus AGT at 5% volume feed on days 5, 7, 9, 11, and 13, and are able to deliver material for really whatever is needed to complete a research workflow. At this point, we've actually done very minimal process development in stirred tank bioreactors. Something we would normally think of as the workhorse for generating our POC material. We haven't had that need at this point. There have, however, been efforts with our development colleagues to grow these cell lines in our Sartorius Amber 250s. We've demonstrated the ability to grow these cells successfully in the ambers to be able to screen some growth conditions in those systems. So we're really looking forward to really putting everything through its paces once we've moved some of these projects further forward. And we've also demonstrated the ability to get in excess of 100 million cells per mil in a high-density TFF perfusion system. So just in terms of some experimental systems that we've tested, the cells have performed very well in really what's been a fairly straightforward system without much additional process or media optimization. So again, these are areas that we're excited to move into in terms of our our process development phase. But at least for my group in the early research side, basically culture, cell line development, and early POC material generation, we've been very successful with using this line. To give you some real world examples of the kinds of things we've done, I wanted to provide you with real data. Obviously not going to tell you what these proteins are exactly, but I'm also not going to just show you our best of the best and say that's what we do all the time, right? There's a very real distribution here, and some of these clones represent things that we did at different stages of our process optimizations. So clearly we're still optimizing things as we go. What I do want to say is that in this case we have seen some good performance in terms of antibody titers. Not everything is a rock star, but we have seen some that do pretty well. And again, mind you, these are canine monoclonal antibodies, and so the differences between human monoclonals performance and say mouse monoclonals, for some of you who may have experience there are just some intrinsic differences that the different species brings you in terms of how stable, how productive you can get cell lines. So this is representative of the types of things we've seen. In terms of FC fusions, these include sort of reagents, tool compounds, and things as well. I Trust me, I would love to tell you that that the ridiculously high producing line that we have is from is a therapeutic that we're gonna go to market with, but that actually is a tool compound. Um, and we have done a small number of sort of non FC fusion, non antibody proteins. In many cases, these present their own unique problems. In some cases, have very high glycosylation states. In some cases, are very small proteins and don't quite express as well. So there's a distribution here, but ultimately, the nice part is, we believe that we're getting good performance, reliable performance out of this, and that we still think that there may be room to improve at multiple stages in terms of potentially our plasmids, our media, our transfection methods, etc. So we're excited about the continued improvement of this system, but also very, very happy with where we're at right now. And for us, we didn't come from really from a point of trying to compare this to a previous system and trying to ask ourselves questions about sunk costs and our how it compared to previous things. The system that we built from scratch, and we made these decisions centered around some key technology platforms, and the CHO-source cell line was one of those, and I would say that at this point that really has enabled us to build our capabilities around that line, and we're very happy with where we're at. And with that, I want to be sure to acknowledge the support and contributions from a group of people, starting with the group at Elanco Discovery Biology. So Crystal Paulson is our cell culture scientist who was involved in the very beginning of the HC Bio P3 cell line evaluation and has continued throughout this entire journey. She's transitioned into being our Beacon power user and sort of our critical cell line development guru, and has been an integral part of developing our workflows with the cell line. Kayla Bean is our cell line engineering postdoc within the group. Although I haven't presented much of what she's currently working on, she was also involved in some of our early assessments of selection methods with the cell line, and continues to be involved in sort of our future state engineering projects down the line. Heather Ringham is another cell culture scientist. She's been primarily involved in the optimization of our transfection and assessment of our transfection systems, as well as our sort of scale up and some of our testing of pools and cell lines. Jared Patch is actually a research scientist in our discovery virology group, but he played some critical roles early on in our assessment of the CHO-source cell line. So I have to give him some appreciation for helping us kick the whole thing off. And then lastly, John Snyder's our discovery biology group lead, who's been very supportive of our licensing of all these different technology platforms. Really early on saw the value of bringing on high quality platforms to build out our capabilities, and has been just a great leader in that space, having the vision to bring all this together. And then really sort of what I view as our cluster of great industry partners. Clearly Horizon has been very supportive in bringing me into this webinar, but throughout the entire process, has acted more as a partner with us in this as we've built out our capabilities. And it really hasn't just been them. Berkeley Lights with the Beacon platform, Demetra with the piggyback transposase system, Adam, not only with the leap-in transposases, but also helped us build out our expression vector plasmid, sort of our Elanco vector series was built, really with their support. So, for me, for our team, building out from scratch, with working with great industry partners, like these guys, it has been a fantastic experience for me, and I have a great appreciation for everything that they've brought to the table in working with me and my team, and helping us really build what I think is a world-class, upstream mammalian bioprocess all the way through our cell line development workflow. And with that, again, one last thanks to Horizon, and I'm looking forward to any questions you may have.
Really nice presentation, Alan. Thanks for showing us the capabilities and power of the CHO source cell line. We appreciate that. For the audience, just want to remind you once again, the Q&A is going to begin in just a few minutes. Send in your questions for Alan and Mario so we can have a very lively discussion. It's really easy. All you need to do is click the Ask a Question panel on the right-hand side of your screen, type in your question, and hit Submit. All right, I want to thank those audience members that have already sent in questions. We have a bunch lined up and ready to go, so that's great. For all the rest of the audience members out there who are thinking about sending in a question, don't hesitate. Type them in, hit send, and we'll get to them as much as we can. Okay, bear with us for just a moment as we transition into the Q&A session.
All right, everyone. We have a bunch of questions that have come in, so let's get straight to them and try to get through as many of them as we can. Alan, first question for you. One audience member would like to know, what are the main differences and similarities between a CLD process for animal health and for human health?
It's a great question, and really, I mentioned, at least in the U.S., the difference in terms of regulatory path, where there may be a different agency that we're dealing with that may be looking for different types of things. And I mentioned that there's one monoclonal on the market now. There's at least one other that's sort of on its way from another company. As far as I can tell, really, the pathways are very similar. The requirements are very similar, and I believe as you get outside of the U.S., the regulatory environment is going to be really identical, if not identical, to what we would have to encounter on the human pharma side. So this is definitely one of those cases where the bar for us is very, very similar, if not the same in some places, as it would be for human pharma.
So as a follow-up to that, the next question asks, do you think the same CLD process you have in place could be used to generate a biopharmaceutical by human health company?
So I believe when we talk in terms of process, in terms of platforms and tools, yes, I believe everything that we've built that in an image of a human pharma company. I would say, as much as I would love to be making human pharma products on the weekends, all of our licensing agreements are specifically for animal health. So that right now is the only limitation. But to directly answer that question, I think that the process we have in place is ultimately identical to what I've seen in a lot of other human pharma companies.
All right, thank you, Alan. Mario, a question for you. An audience member would like to know that if they license your platform, what is the level of freedom that I have to change protocols and to use these cells for other purposes?
Okay. So basically, as you could see by Alan's presentation, the level of freedom for changing the process is quite big. So we don't restrict pretty much any change. The protocols that I've mentioned that we supply in the beginning are a good starting guideline for you to develop CLD process, but ultimately, all the years that you have already gained throughout the time, all the experience is not going to waste, so you can use a lot of your knowledge on it. So transaction selections are processes are, you can change them. Medias and feeds as well. It's possible to change them, and of course vectors. So it's not a thing. So this is from a technical point of view. And another thing is we typically support those changes if the customer comes to us. So that's one of my job roles as a field application scientist, is actually to support customers that are willing to do some technical change. In terms of other purpose, I think the licensing terms, you can use it for manufacturing, but you can also use it, for example, for early discovery phases. So you can use it, for example, as on your discovery pipeline as well as this meaning from your internal R&D, providing, of course, that you have a commercial license for manufacturing. And also, I think there's no many restrictions on the CMO that you partner with for production. So it's actually quite a flexible platform on that terms as well.
Great, thanks, Mario. Alan, next question for you. Did you compare transposon systems for same molecules, and if you could share your results?
So we did not in our shop compare sort of the same molecule side by side, head to head. I know there have been others, and there's I believe a publication by some of the team at Lilly that looked at efficiencies, but ultimately in our hands, what we had been using the piggyback system sort of as a part of our I guess you could say legacy workflow, and continue to use that now. The big attraction for us to the leap-in system initially was for some of our engineering efforts, the ability to have a transposase that would not interfere. So it's something we could utilize a transposase on a line and then come back in with a different transposase at a later date. And in the case of leap-in, there's actually two different leap-ins. So in theory, if you have access to all three, you could potentially imagine sort of three different rounds of transposase integrations that would not interfere. But at least at this point for us right now, the leap-in system we've been using for some of our engineering efforts and exploratory efforts, and then the piggyback system has been what we've used for our therapeutics. But I believe that they function very similarly.
Great, thanks, Alan. And next question is for you as well. It starts off by saying that in your last slide, you show some good expression data with different molecule modalities, but specific for canine production, I believe. In your opinion, do you think that expression capacity would change if you were producing a human monoclonal antibody or human Fc fusion protein?
So that's a great question. I have limited data for that. I would say that in my opinion, I think that there's always going to be a ceiling, that you'll hit at some point, and that's in many cases a little bit process dependent. I do recognize that there is some molecule dependency there. In what limited experience I have with this system, with a human antibody or human Fc fusion. And granted, this is not something that we have done all the way through to clonal cell lines. These are things we might have made as a tool compound, and looking at pool titers. So I want to be clear on that. I would say that in those cases, those have been slightly better performing pools. Given the small n in that assessment, I don't believe that there is some order of magnitude difference there. So I think the performance between canine and human antibody or Fc fusion proteins is probably similar in these systems. And in our experience, the couple that we have done have been slightly better at the pool stage.
All right, Alan. And why do you select the 40 CHO Dynamis as the primary medium? Because of higher lactate production from the cells?
So ultimately, the original selection of Forticho was based on a recommendation from Horizon. The cells, as I mentioned, they seem to be quite prolific in the Forticho media. What we've, I'm not on the team that's doing the media optimization. That's a gentleman by the name of Brian O'Reilly in our development team. But the work really has centered around sort of the changing richness of the media, I believe, and switching that around from sort of standard maintenance to N-1 all the way up into production. To my knowledge, we have not necessarily at this point used lactate production to determine whether or not we would use media. Ultimately, it was growth and transfectability. So I don't know if Mario has any other comments on that on his experience.
Yes, actually. The choice of CD40 as it's a bit of an historical one. So the cells, when they were adapted to suspension, they were actually adapted to CD Opticho initially because to give quite a broad adaptability and flexibility to other medias. And then we then typically recommend CD40 because of our recommendation of the lipid transaction method as one of the alternative methods. One of the markets is new biotechs or new companies that are still building up some labs, and so you can easily buy a Freestyle Max, for example, vial to do your lipid-based transaction as a starting point, and then you move on to electroporation. And CD Opticho was not compatible with lipid transactions, that's for the main reason why the Forticho. But we know that our cells grow quite well actually in a variety of the shelf medias. So we have customers working with XL CDCHO fusion from Sigma, the Balance CDCHO from Irvine, the iClone ActiPro from Cytiva. So it works quite well in different basal media as well as other feeds. So, for example, Alan showed the efficient feed C+, but we know that we have other people using Cell Boost 7A and 7B, and the feed 4 from Irvine, for example. So it's quite a robust and quite a flexible cell line on that regard.
All right, thank you, gentlemen. Alan, back to you. What level of expression were you making from the lipid-based transient transfection method you tried out?
So it's been an interesting exploration into this, and I honestly wish that we had more time. Although I don't know if the people who are actually doing the lab-based work wish we had more time for this optimization. But I've been really kind of fascinated by the fact that something like Freestyle Max, which has a relatively high transfection efficiency, not quite as high as we've seen from something like the Neon, but seems to do fairly well. And actually, in terms of the data that I showed, most of those pools and clones and things that I've talked about were done with Freestyle Max. And it hasn't been until a little bit more recently that we've moved to the Neon system as sort of our standard. And as I mentioned, with the limited data set we have now, the initial sort of at the pool level with the Neons, or sort of the post-transfection efficiency viability seems to be better. After we get out of selection, we're seeing very similar performance, and so I hesitate to say that across the board with the fewer numbers of cell lines and pools we've done from the Neon system, but comparatively, the Freestyle Max has done pretty well for us.
All right, great, thank you. Mario, we have a question for you. An audience member would like to know, what are the usual production titers by using CHO source cell line?
That's That's quite of a broad question, to be honest. Because it's not just depending on the cell line, but it's also there's a lot of other factors involved in it. But we have reports of good pools expressing between the half a gram per liter, but a bit also 2 grams per liter, for example. And then the clones, they can range all the way from the 2 gram per liter all the way. We have seen a situation recently where a clone was expressing 8 grams per liter from a report of a customer. So yeah, it's quite broad. Of course, it depends if it's an IgG. Sometimes it's with a standard IgG. It's quite easy for something as simple as, for example, trastuzumab, or if there is something a bit more engineered, or with an Fc fusion or an engineered monoclonal antibody. Yeah, it's a bit because sometimes it's a bit lower, but the ranges are what I mentioned before. Yeah.
All right, thank you, Mario. Alan, in your Beacon Spotlight assay, you showed a distribution on productivities. Was this pulled from an integration approach, such as piggyback or leap-in, or was it a plasmid transfection?
So the data we showed was that was a piggyback transfection, piggyback integration that we had done. I will say that distribution was somewhat, because we ran that sort of as a more of a proof of principle that was fairly early on in some of our protocols. So a lot of that had not been optimized yet. So I think one of the things that the attendee is asking, and that it's an interest of mine as well, is can because we can generate this sort of distribution data on the Beacon, can we use that to infer efficiency or distribution, or are there other other information we can pull from that to optimize? That's a question I've had as well. I think that this is one of the inherent problems in building the airplane as you're flying it, which is we have not done as many runs where we've systematically done things exactly the same every time. And so it's really hard to get a handle on how much some of these different variables have impacted our distribution of clones. And so we're even still working on optimizing our Beacon Spotlight assay, because the catalog Spotlight reagents from Beacon are all for human and mouse. And so we've had to working with Berkeley Lights, who again have been a great partner alongside of us on this, have worked with us to develop a canine-specific reagent, and so we're still making improvements on the reagent and the assay as we're going. So I know that's probably not a very satisfying answer, but yes, it's a very interesting question and something that we are looking into.
All right, great. And Alan, do you see any advantage in pursuing a perfusion manufacturing process over a fed batch process for reduced cost of goods?
So I could probably go on for a while about all the pros and cons and the interesting things going on in terms of fed batch versus perfusion, and I won't bore people with my sort of opinions and things on that. But I would say that I think in animal health, again, we're confronted with some different challenges from human pharma, and in terms of cost of goods, in terms of even peak annual demand for material. We're not looking at products that are going to require hundreds and hundreds of kilograms of antibody per year. And so when we start getting into questions about early capital investment in manufacturing footprint and all these other kinds of things, I mean, there are much smarter people than me that model all this stuff out, and I find it fascinating. But I think that's definitely a question that we're asking. It's also interesting that for people who may be a little familiar with how the Beacon system works, the Beacon is essentially a microfluidics perfusion system. So I'm not going to say it's analogous to a true perfusion system, but that's another one of the questions that I've had is, as we build these things out, if we move into a perfusion process, are we actually already stacking the deck in favor of that because of how we're selecting our clones, and that we are selecting not only high-producing clones, but clones that would even, that would produce well in a perfusion environment in addition. And so, all good questions. I'm excited about the prospects for perfusion actually across all of biopharma. I mean, I think that there's some really great niche sort of boutique biopharma applications that could benefit from that type of strategy. So I'll leave it at that.
Great, thanks, Alan. Mario, question for you. Were the media and feed used recommended by Horizon?
If that question on Alan's presentation, I believe so. Alan, can you correct me on that one?
Yeah. So the ones we used, we started out using because they were the initial recommendations by Horizon. I mean, as Mario mentioned, we're aware of other medias and feed systems that people have used in addition, and honestly, we've tried a couple other different feeds that work well, or that at least in some initial tests might have worked well. We're satisfied with where things are going. Again, I have a little bit of a luxury of being on the early research side, and it may end up being that as we build out and more fully develop our development component to this and manufacturing, we may revisit what we do on the research side for that. But ultimately, we started off with their recommendation. The cells grew really well. They were very healthy, transfectable, and that were our key criteria. So we didn't explore much beyond that.
All right, thank you. And Alan, I think we have time for one last quick question. One of our audience members would like to know, could you share what type of scale or flash size was used for titer testing slide in your slide number 28?
Yeah, so most of those were done in 125 ml shake flasks. A couple of those, as I mentioned, that we sort of did some side by sides between 125 ml shake flasks and then these 24 deep well plates. As we've shifted into screening larger numbers of clones at a time, it's been better for us to work on the 24 deep well plates, and so a couple of those titers that were on there are actually from the 24 deep well plates. But like I said, we did some comparability studies, and really the titers are predominantly plus or minus 10% from between the two methods.
Alrighty, thank you, Alan. And with that, we've come to the end of our webinar. I'd like to remind everyone that the webinar will be archived on the GEN website for up to a year. That's at genengnews.com. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends or colleagues. I'd like to thank Alan and Mario again for their very informative presentations, and I'd like to thank you, the audience, for your attention and thoughtful questions. And a very special thanks to Horizon Discovery for sponsoring this webinar. So hopefully we'll see you again at another GEN webinar in the near future. Goodbye for now, and everyone stay safe and healthy.